Start. Hello, everybody. Oh, one more person.
Hi.
Hello.
Hello, everybody. Welcome to our webinar. Uh, we want to show you the data analysis platform that we have developed and to show you how easy it is to analyze sequencing data using this platform. My name is Artem Barski. I am associate professor at Cincinnati Children's Hospital and co-founder of the Ethereum. I joined Cincinnati Children's and started my lab about 10 years ago. And my research interest is how does the epigenome determines the memory status of T cells. And in our work, we use a lot of next generation sequencing based data such as ChIP-Seq, RNA-Seq, ATAC-Seq, and so on. And of course, we needed to find a way to process this data. So my first hire was actually a bioinformatician. So he was processing the data, and usually it would take him some time. He needs to focus on one project, he needs to focus on another project, and then the data are sitting there unprocessed and everybody is unhappy. So what we decided to do is to develop a platform that would allow biologists in my lab to do initial analysis of the sequencing experiments and to decide, for example, did the experiment work or did it fail? And this is where SciDAP comes, and this is actually a second generation of the platform. The first one was called BioWardrop, and here are some of the features. So first of all, it is designed to be used by both biologists and bioinformaticians. You don't need to know R, you don't need to know Python, you don't need to know Linux, you don't see black screen, but you still can process the data. For bioinformaticians, it offers the convenience of once you establish the pipeline, or you can use our pipeline. So all the initial processing will be done by the system, by people who develop the data, and you don't need to rerun same boring pipeline every time. You can do something more interesting like post-processing of the data, making some biological conclusions. So what the platform does, it allows biologists to perform initial data analysis by themselves. And this way, the results can be obtained faster. You get push button analysis for many current methods like RNA-Seq, different versions, ChIP-Seq, ATAC-Seq, CUT&RUN, CUT&TAG. We are also including single cell data as well as methylation data. There is matching post-processing analysis, for example, differential expression for RNA-Seq, binding site and Heatmaps, and you can also integrate ChIP and RNA data. And we keep updating the tools constantly. One big advantage of SciDAP is that it follows principles of fairness as demanded by NIH, in that both data and the analysis are findable, accessible, interoperable, and reusable. So the data are centrally stored. People who are working on different projects can work with the same data. And the pipelines, the way we run them, are very reproducible thanks to the use of pipeline language called Common Workflow Language. So in summary, for biologists, we provide fast turnaround of the analysis. So you can know whether your experiment worked within just a few hours. For bioinformaticians, we allow to easily handle pipeline, modify them, easily provide visualization tools. And for everybody, we make it much easier to handle the data. You don't need to convert files. You don't need to run around with hard drives. All the data are stored together, well annotated and reusable. So this is just a quick view, and we will go in the system itself in a few minutes. But just a few screenshots. This is how you start analysis. You need to select what experiment you did, ATAC, for example, or ChIP-Seq, provide the name of experiments, conditions, and genome type. Then you attach file with the data. And once you click save, the system will process. You will see something like this, where we have samples. And this is the list of all the samples available in this particular project. You can, for example, see what is the mapping statistics here. You also get pretty detailed QC data about your experiment. You can look at various QC plots, for example, this base frequency plot, or you can look at your data on the browser. So this is RNA-Seq. You can see that there is clear intron exon structure, but you can also see that there seems to be a loss of signal on the 3' end of the gene, suggesting that RNA might have been slightly degraded. You can compare different samples on the genome browser. So for example, here we have two histone marks, histone H3K4 di and trimethylation, ChIP-Seq, and also ChIP-Seq for RNA polymerase II. And you can see that where they bind, and you can compare the binding patterns. So these are easy to use features for people who don't program, but for people who program, we offer a very easy addition of pipelines with common workflow language. Common workflow language pipelines can be easily imported into the platform if you want to add something new. And we are happy to help people develop their pipelines. And the great part is that this analysis is reproducible because all tools are containerized, and data processing can be performed on any hardware, from your desktop to your server to cluster to cloud. So a little bit about hardware. So SciDAP contains two parts. One is our central server, SciDAP.com. This is where you log in to access the interface. This is where you register. But the actual data processing are done on what we call satellites. And satellites can be computers such as Mac Mini with sufficient amount of memory and processing capabilities. And all the data processing is actually happens on these satellites. And the satellite is something that the client laboratory owns. So the data actually never leave the satellite, and they stay within your local network if this is where your satellite is. The only communication is with SciDAP, because SciDAP would be giving instructions on how to process the data and also provide the interface for viewing the data. So the advantage of this is that your data are always with you. Nobody has them, but also you get access to the latest version of user interface and the latest version of pipelines, and you don't need to update anything. You will always have the latest interface. Okay, we still have people coming. Welcome. For bioinformaticians here, I would like to say a few words about the common workflow language, which is what all our pipelines use. So common workflow language is a way to describe the processing which is easy to understand and easy to reproduce. So there are two elements in Common Workflow Language or CWL. One is tool and another is workflow. So tool is a single command. For example, let's say you want to map the reads. So in CWL, we describe what are the inputs for this program, what is the command for this tool, and also what is the operating system environment. And this we provide as a Docker container. Also, we describe what are the outputs of this tool. And because inputs and outputs are so well described, it is easy to connect the tools into pipelines. And in fact, there are even graphical editors that allow you to just snap tools and pipelines, snap tools together into a pipeline. So our know-how is what we call CWL extensions. And this is what makes SciDAP so user-friendly. So in addition to the pipeline instructions, we also include information on how to create the input form, as you saw previously, what is the pipeline, and how to present the results. For example, pipeline produces coverage file, so this needs to be displayed on the genome browser. Pipeline produces differential expression data, so this can be displayed on Volcanoplot. And so our pipeline tells the system how to display the data, and basically this is kind of like old style gaming system where you change cartridges and it produces you get different interface. Okay, so let me show you actual pipeline then, and I'm sorry, actual platform. So this is our website, scidap.com. And if you want to register, you can click here. And here you can register, and this will give you access. We are updating our platform very soon. And so actually tonight, and when you log in, you will see something like this. And this gives you an option to choose how you want your data to be processed, and whether you want to use your own satellite or you want to use our satellite.
Hey, Artem, I think we're still seeing your slide deck. Did you?
I'm sorry about it. Let me change the sharing.
There you go. You might want to explain again what they're going to see, what you're reporting to.
Yeah, sorry about it. So this is scidap.com. This is start trial button. We provide a 30-day free trial. If you need a few days longer, we're happy to oblige. And this is the screen that you will be seeing starting tomorrow. So here you will have a choice of either join existing laboratory, start a new laboratory, or start a laboratory with a trial satellite. So the easiest for the trial is this one, start trial laboratory. This will connect your laboratory to our trial satellite, and the processing will be done there. But if you decide to continue using SciDAP, we recommend to actually use your own satellite computer for data processing. You will need something something like Mac Mini if you are working with human or mouse or other mammalian genomes. And if you're working with Drosophila, even a laptop will do. And finally, if if your PI or other lab members of yours have already started laboratory, you can say join laboratory, and the admin the administrator of the laboratory can send you an invite to join. So let's look at actual data analysis. So this is the dashboard that you will be seeing. And in SciDAP, the data are organized into laboratories. So for example, this is my academic laboratory, and this is a trial laboratory to show today. And within laboratory, we have projects. So the first thing you would need to do is to start project, and you can do it by clicking Add Project button. So here you can provide some description of the project. And the next important step is to attach the pipelines that you are planning to use. This way, all the data sets that are part of this project will be processed by the same pipeline and will be comparable. So for example, if we want to do single cell data, we can select, we can, for example, look for CellRanger Pipeline. And here you can put a check mark, and this will add the pipeline to the project. So after you set up the project, if you need to add additional pipelines, I'm not going to save it, but you may need to click the save button. After this, you can edit the project and add additional pipelines by clicking this three-dot menu and selecting edit, and you will get the same screen to add additional pipelines. So let's look at the project, and let's look how we are adding the data. And I'm going to show you the data from our recent Journal of Experimental Medicine paper. In this paper, we looked at activation of T cells, and we have ATAC data in resting and activated T cells. We have RNA-Seq data in resting and activated T cells, and we have ChIP-Seq data for Jun transcription factor, among others. And this is what we're going to look at today. So let's start by adding the data. So the button to add a new sample is new sample. So here you can select a pipeline, for example, ATAC pipeline, and you provide basic description here. You select the genome, and you need to provide the FASTQ read data. And we provide several options for doing this. One, you can upload the data from this computer, from your current laptop. So select the file or drop the file here. Or alternatively, you can add a file from FTP site by providing an FTP link to the file that your core facility has created. If these are not your data, but the data from the database, then it is even easier. You can just enter the accession number, and here is you enter GEO and SRR number, and the system would automatically pull this data and make it easier for you. You don't need to look for files. So once the data are added, you click save, and the system would probably take a couple hours to do the processing, but this is what you are going to see. So let's look at this one, for example. So the first screen is quality control. You can see the mapping statistics. You can see what is the percentage of the reads that are mapped or unmapped, what is the adapter, what is the number of peaks that have been called in this experiment. Next, you can look at the data on the genome browser. It takes a few seconds to load. So here we are looking at actin B gene. And you can see that there is a peak around this gene. But let's also look at, you can look at any other gene you like, for example, IL-2 here. And you can see whether it has signal or not. You get quality control plots. So just to explain, this one is base frequency plot. As you know, human and mouse genome are AT rich, so you see more A's and T's here than G's and C's. This is the base quality plot. This is what is provided by Illumina. So this plot shows you the enrichment around the transcription start sites of all the genes. So we have open chromatin at the promoters of expressed genes, so we see a peak there. And we also have a very cool feature called pre-seq estimates. So this is something that allows you to determine whether it is worth getting more sequencing. So in this case, in this experiment, we had, let me see, we had 33 million reads. And out of them, we had a certain number of unique reads, so probably somewhere around 20 million unique reads. So if this plot allows you to estimate how many unique reads you will get if you sequence more. For example, if you sequence 100 million reads, then your total number of unique reads will be about 32 million. So you probably want to stop sequencing somewhere around this point where the curve bends. In addition to the QCs, you of course get the list of peaks, and we provided together with coordinates of the nearest gene. So here, these 3 columns are the coordinates of the peak. Next to it is the what is the nearest gene to it, and also you get what kind of area is this? Is this upstream of gene? Can it be considered promoter or maybe intergenic region? And also you get P and Q values for the significance of the peak here. You can save this data by right-clicking, and you can export the data as Excel or CSV. Or also all the files that are produced by Pipeline are here, and you can download both the log files and the results files. Okay, we have actually ATAC data in triplicates, and what we want to see is whether there are changes in chromatin accessibility that are induced by T-cell activation. So one easy way to do this is to just look at the data on the genome browser, and we have this tab where you can compare all the data. You can select the data that you want to look at by just putting this check mark, and this would add the data to the browser window. And you can compare the peaks between different samples and experiments. Takes a second to load. Okay, let's look at the gene which we know is changing. So IL-2 gene. So if we zoom out, so here on the top 3 tracks are in activated cells. And here you can see that there is open chromatin area both at the transcription start site of L2 here, as well as at this upstream enhancer in all 3 of our activated T cell samples, but we barely see anything in our resting naive T cells as expected. But of course, this is just a visual analysis and it's good to look, but we also provide an opportunity to do more statistical analysis. And here we have another tab which allows to do comparative analysis. So if we want to compare ChIP-Seq or ATAC-Seq experiments, we use a tool called DivBind. So in order to do this, we select the pipeline, provide a name for the experiment, and then select what are our conditions. So we just put check marks next to the experiments we want to compare. Naive, activated, check marks. We can give the name to the sample. So for example, in this case, resting and activated. And after this, we just need to click Save Sample, and the system will run the analysis. Probably will take about half an hour. And this is how the results look like. So again, you can look at the results on the genome browser. And I will go to my favorite IL-2 example. So here you can see the top part is the signal. The bottom part are the peaks that are called in each of the 6 samples. And the green shows the samples where, or red, if it is in the other direction, shows the peaks which are differentially accessible in different samples. So for example, these 2 regions are differentially accessible, and you can see what is the p-value or FDR for the difference. On the next tab, you can look at the same data in the table form. So this gives you what is the peak, what is the nearest gene, and as well as what is fold change and p-value for the difference. Finally, the pipeline produces a number of plots. So this one is overlap between the samples, and you can see that the 3 activated samples are more similar to each other than to the resting samples. This one looks at peak count correlation, and this Venn diagram shows you the number of samples which are in common or separate between naive and activated cells. And as you can see here, when we activate the cells, there is a lot of chromatin opening. So out of 11,000 peaks that are common, we get another 15,000 peaks which are present only in the activated cells. You get heat map, and importantly, you get a PCA plot, in which case, so you can see how different your samples are using either all sites or only differentially bound sites. And you can see that the first principal component contains 50% of variation. So it separates resting from activated cells, and the second component separates the donors. Do you guys have any questions about ATAC-Seq analysis? If you want, you can type them in the chat or just say it.
Yeah, everybody's on mute, so we won't be able to hear you. You have to take yourself off mute if you have a question.
Okay, so we see these wonderful changes. So next, we would like to see what are the transcription factors that might be driving this. And we actually did this analysis, and we found that it is likely to be AP1 transcription factor. So next, we did ChIP-Seq for AP1 component called JUNB. So again, to add a sample for ChIP-Seq, we also select ChIP-Seq pipeline, and we'll again provide the same data as before, including the accession number or the link to the FASTQ file. And when the data are processed, you will see the experiment here. And you will see the basic statistics, number of peaks. You can look at the data on the genome browser. So you can see there is a lot of signal at actin B, but let's look at our favorite IL-2. Here you can see at least a couple of JUNB binding site next to the IL-2 gene, suggesting that it's regulated by AFOS, which is good, but we would probably like to make sure that our JUNB chip worked. So for this, what we can do, we can look at the peaks that have been called here. This is a table, and we can select the coordinate of the peaks that we like, and then we can use this in motif analysis program. So what I did, I took these coordinates, and then went to a program called Homer Motif Analysis. And I provided these coordinates as a CSV file or BET file. And then I was able to identify the peaks, the motifs that were enriched at the JUNB peaks. This is how the results would look like. You would actually get only this files pane. And here you can open the de novo calling. So these are the motifs that were identified by Homer de novo analysis. And the top motif is this TJ and TCA. And this is a motif which is similar to FRA1. This is a member of AP1 family. They are all binding to the same motif. So this suggests to us that ChIP-Seq for JUNB worked, and we're getting the correct motif. So now let's go to back to the project, and we are going to look at the RNA-Seq data analysis, unless you guys have questions about ChIP-Seq. Okay, so for RNA-Seq, it is also very simple to add new data. So you use this new sample button again, select appropriate RNA-Seq pipeline, and we have several versions for this. So the data can be strand specific or DUTP method, or not strand specific, and they are processed slightly different. There are also libraries prepared by different methods. So one, there is Illumina or NEB kits. So these are the ones that I used more often. There is also SmartSeq from Takara, and this requires a different pipeline, even though it is strand specific. And finally, there are, you can sequence the libraries as single read or paired end, and you need to select appropriate type of experiments. Again, you provide the information about the cells, about the genome, and you need, again, to attach a file or provide a short read archive accession number. Once you click Save Sample, it will be processed, and this is how the result would look like. So let's look, for example, at this one. So this is the statistics for mapping, and the green ones are the reads that are mapped to the transcriptome, and the orange are the reads that are mapped outside annotation. So this might be a DNA contamination if you are having too much of it, or this might be a transcription that has not been annotated. You get the number of mapped reads here. There is a very useful statistic, which is the number of reads that are mapped to ribosomal DNA. So this can tell you whether your ribosomal RNA removal was successful or not. And as well as other statistics. You can look at the data again on the genome browser, and you can see whether you indeed have the expected exon intron structure. Okay. Yeah, so this is actin B. You can see this is a strand-specific experiment. You can see that this gene is transcribed from right to left, and we see that the reads are actually mapping on the opposite strand, so on the top strand rather than bottom strand, which is expected, and suggests that our DUTP strand selection worked. There are some QC plots, and I think this one, base frequency plot, is quite important. As you remember, human genome is AT rich, but the genes are actually not AT or GC rich, and you have the similar percentage of each base. And unless you have a contamination with DNA, you should be able to see something like similar to what you see here. Here is a table of gene expression. So these are RPKM values that we're providing. So this is normalized and can be compared between samples. We also provide not normalized total reads here. And we actually calculate gene expression 3 ways. So first, we start by calculating expression for each gene isoform. And then we sum up gene expression for isoforms that start at the same promoter. We call this common TSS. And then alternatively, we provide values for the whole genes where we sum up expression for all isoforms. So for example, here we have 3 files: RPKM for isoforms, RPKM for common TSS, and RPKM for genes. So if you are studying activity of the promoter, then probably you want to use the common TSS. And if you are studying more like function of these genes, then the gene-wise values will be more useful. Okay. So let's say you edit your experiments, you are happy with them, and you want to do differential gene expression analysis. Again, you go to the comparative analysis, and you can look at differential expression. Here we're looking at naive versus activated T cells. So to do this analysis, you use this new analysis button, select the program. We're using DSeq, and you can select which experiments you want to compare. So we put check mark at naive versus activated here. We can name the conditions, naive and activated, and click Save Sample to start the analysis. It is about 15 minutes. So once it is completed, this is approximately what you will see. So of course, you want to see the table. So this is for each gene. You can see average RPKMs for naive and activated, log fold change, and adjusted and unadjusted p-values. You can see right here. So you can filter, use advanced filtering, which is pretty self-explanatory, or you can download file and filter it in Excel to identify genes. We also produce volcano plot. So here you can see the genes. We are plotting log fold change versus log p-value. And so these are the genes that are probably more changed. So you can highlight, see the name of the gene and the values. Other plots that are produced by these pipelines are here. So this is MA plot. So this is log fold change versus log geometric mean. And the blue ones are the ones that are significantly changed. And here we have principal component analysis. Again, you can see that on the principal component 1, which contains 94% of the variance, we're separating naive from activated samples. And the second principal component, only 4% of variance, is the donor difference. And of course, you can download all these images for use in your paper. Okay, so now we did this differential analysis, and again, we want to make sense of this. So we want to take these differentially expressed genes and use them in gene set enrichment analysis to see what do these changes in expression actually mean. So again, we're using pipeline, which is called GCA here. And we just need to provide results, the DSeq experiment as an input. So we have this naive versus activated differential expression. We can select gene set annotation. So for example, hallmark or gene ontology here. And then you click Save Sample, and it will run the pipeline. This one is pretty fast. So this will give you a table of all the different terms that are enriched in your activated or suppressed genes. Or you can look on the data on the graph. So these are enrichment plots and heat maps. So you can download these files. And once they download, you can look at them. Just a second. Okay, so for example, we know that there is IL-2 STAT5 signaling, and indeed, this is something that we see here, and this produces the GCA plot. You can see that the genes that belong to this IL-2 STAT5 signaling pathway are strongly expressed in the activated T cells, as you can see here. You can look also at these genes one by one on the heat map. And so this is the heat map. It includes all the genes that are involved in this pathway. And this shows you the data for fold change. I'm sorry, for changes in gene expression. You can see that many of these genes are induced in the activated T cells. Okay, so we have also a number of other tools, and also the tools for single cell analysis that we are adding to the platform. And I can either answer the questions or show you the single cell analysis if you would like. Any preferences?
Hi, my name is Oh Kyung, and I'm from UT Austin, University of Texas at Austin.
Mm-hmm.
Actually,
Okay.
I had my data, and then I didn't know what to do.
Mm-hmm.
I'm not that expert about biomed this kind of stuff, but I learned a lot from actually I went to the Illumina.com
Mm-hmm.
that website. Actually, I didn't know what to do with my data, but with your explanation and then description, and I can say it. Definitely, I'm gonna try this one. With my data.
Yeah, please, please try. And if you have questions, feel free to contact us. We pride ourselves on explaining stuff.
Thank you so much.
Thank you. Should we stop, or would you like to see a single cell? Okay, let's quickly go over it also. I'll reshare my screen again. Okay, so for single cell, I guess the problem for many people is that the data set is huge, and then you need to quickly reduce it to a relatively small table which contains gene expression value for each cell. So here we simplified it, and to do this, you can use CellRanger Pipeline, which is developed by 10x to process the data. Again, processing is very simple. You select the pipeline, you select the genome, and you attach files. So it is usually paired end. So you can again enter either the FTP link or SRA number. If this is paired end, then you just put the same SRA number for both read 1 and read 2. Once you save the file, you can look at the results. And what we produce is actually two things. We produce the HTML report. So this is just basic quality control. we produce, just a second. This report, which tells you what is the number of barcodes or unique reads per cell, and you can get basic statistics like what is the mean reads per cell, what is the cell with significant enough coverage, what is the number of genes measured per cell. You also get this initial t-SNE plots, which can be colored by clustering or by counts. You can look at some of the genes, but this is not very interactive. So if you go back, the main file that we produce is a CLOOP file. This one is bigger, so you can download it and then use 10x data browser to view the results. So I have actually downloaded it before, so let's. And here you can look at this data in many different ways. You can, for example, switch between t-SNE and UMAP clustering. You can select which clusters to show, which clusters not to show. You can highlight your gene of interest, and so on. Also, if you have more than one 10x run, you can use CellRanger aggregate pipeline to combine two initial runs. So here you can, we actually have only one, so we will not be doing this. But in CellRanger Agg, you can combine several CellRanger processed experiments into one, and this way you can integrate different types of data and see how they cluster together. And again, view the data in the CLOOP, but also in the new version, we will have an updated view, which will make it easier to look at this data. Any questions? Okay, thank you very much, everybody.